Next up is Rickesh Patel, and he received his bachelor's from Cal State Long Beach, down the road from me. He then did his PhD with Tom Cronin at the University of Maryland, Baltimore County, looking at homing in mantis shrimp. He's very recently now a postdoc in Stanley Henza's lab at the University of Lund, studying path integration. And with that, I will pass it over to Ricky.
Hi, let me go ahead and share my screen. All right, so thanks for that introduction. So like Melissa said, I recently graduated, finished my PhD in Tom Cronin's lab at the University of Maryland, Baltimore County. And I'm pretty excited today to share with you a large portion of my dissertation research. So just first, I'd like to thank ISN's Young Investigator Award Committee for allowing me to present today. It's a real honor, especially with the company that I have. Okay, so I'm gonna go ahead and get started. So I worked with mantis shrimp, and these are predatory crustaceans, which are really well known for a few things. First, they have these really wonderful visual systems, which impart in most species. It's color vision, linear polarization in multiple spectral channels, and circular polarization vision as well. They're also really well known for their powerful predatory strike, like you just saw. So most mantis shrimp species live in shallow tropical waters, such as here off the coast of the Florida Keys, where my study organism, Neogonodactylus orystidae, can be found. So there's an individual in a small hole in the seafloor, and most mantis shrimp species occupy these small holes for use as burrows, where they spend most of their days safely concealed from their predators. So these burrows are extremely important resources for these animals. But despite the safety these burrows offer, they will leave their homes occasionally for tasks such as foraging. And these foraging trips can extend up to a couple meters away from their homes, at least in this species. But they will turn around and reliably return to those same burrows, which led me to ask the question, how do mantis shrimp navigate back to their burrows after these foraging excursions? So here are a few examples of burrows that can be found at that same field site where I recorded the video you just saw. And you might notice that the environment is fairly structurally complex. So there are sponges and coral fragments and pieces of aquatic vegetation that these mantis shrimp may be using to aid them in navigation back to their homes. So I hypothesize that they're using these structures as landmarks during navigation. So in order to test this hypothesis, I built these circular navigation arenas. They're essentially kiddie pools filled with sand and seawater, and I placed those arenas in the greenhouse on UMBC's campus. I included a vertical PVC pipe that I buried in the sand so the animal had a burrow to live in. And I included a vertical landmark with black and white horizontal stripes adjacent to the burrow in some cases. So actually, let me pull out my laser pointer. Okay, so if you look at this overhead view of the arena, this white circle is the burrow's location, and this orange circle represents the landmark. Okay, so I also included pieces of food, which I would stuff in empty snail shells, and I placed those food pieces in one of two fixed locations in the arena. So those are marked by these black hexagons here. And I'd observe outward paths that the animals made from the burrow to the food location, and homeward paths the animals made back to that burrow again. And I did this initially under two experimental conditions. The first in which the landmark was present adjacent to the burrow, and the second in which the landmark was absent. So here's an example of a trial in which the landmark was present. So this blue path is the animal's outward path that's emanating from this dark circle, which is the animal's burrow. Adjacent to the burrow, you can note this white circle is the landmark viewed from above. So you can see this outward path to the food the animal's making is fairly tortuous with twists and back turns, and it's very slow as well. But you'll see once it finds the food in a second, that the animal makes this homeward path that's quite different. It's very direct back to the burrow's location. So that was really exciting for me to see. And here's the path tracings of the video you just witnessed. So these blue paths are the outward paths away from the burrow to the food, and the red path is the homeward path back again. So looking at the homeward paths of multiple individuals, you can see that in the presence of the landmark, mantis shrimp's homeward paths were very direct towards the burrow's location. Okay, so next I wanted to see what would happen in the absence of the landmark. So in this trial, the outward path is in blue, and you can see that there's no landmark adjacent to the burrow. So the path sped up, but once it finds the food, it'll be in real time. Okay, so the animal's now found the food and it's in real time. You can see that the homeward path is fairly direct near the location of the burrow before the animal turns more than 90 degrees from that original homeward trajectory. So that's when I decided the search behavior would be initiated, and the animal continues searching until it finds its burrow. Okay, so there again is the tracing of the path you just saw. And when I did this with multiple animals, you can see that in the absence of the landmark, animals made these fairly well-oriented paths towards the burrow's location, but when an animal did not find its burrow after this initial homeward trajectory, it would initiate a fairly extended search behavior. And this is in contrary to when the landmark was present. So you can see that here, and these homeward paths in the presence of the landmark were much more direct. That's pretty much due to the virtual elimination of these extended search behaviors, which were instead centered around the landmark's position. Okay, so I measured the orientations of these initial homeward paths in red in both the presence and the absence of the landmark. I have a lot of these orientation plots in my talk, so I'm just going to guide you through them really quick. Each of these points on the circumference of the circle is a homeward orientation of one individual in relation to the direction of the burrow, which is the triangle above the plot. And these arrows in the center of the plots are mean vectors. So the direction of those vectors are the mean direction of all these points, and the length of the arrows indicate how strong the orientation of the population is. So, so you can see for these initial homeward path orientations, animals were fairly well-oriented towards the burrow in both the presence and the absence of the landmark. These were measured at about a third of the beeline distance, or the most straight path from the food location to the burrow. So right around here. However, if I measured the orientation of the homeward paths right before search behaviors were initiated, so I termed those the final homeward path orientations, you can see in the presence of the landmark, these homeward paths were very well oriented to the burrow's direction, and this didn't happen when the landmark was absent. So it appeared that in the presence of the landmark, animals corrected for any error that they had in their initial homeward orientations. So it appears that they were using the landmark when it was available, but this doesn't really explain how mantis shrimp were able to orient towards their homes even when the burrow was unmarked. So many stomatopods are central place foragers, and a central place forager is an animal that returns to some sort of central location between foraging bouts. So other central place foragers in nature have shown to be able to use a navigational strategy called path integration during navigation. So during path integration, an animal keeps some sort of reference location, like its home, and continually monitors the directions and the distances they travel away from that starting location to, at any point in time, calculate the most direct path back to that starting location. So this homeward path, called the home vector, is continually updated along the course of the outward path. And this home vector is composed of both the direction the animal should travel to find its home and how far the animal should travel before reaching its home as well. So in order to see if mantis shrimp were using path integration when the landmark was absent to locate their burrows, I built a track with a platform that could be slid along the track. I would place food on that platform and let the animal find the food. Once the animal found the food, I would displace the animal on the platform to a new location in the arena, and I would observe the homeward paths the animals made. If animals made a homeward path parallel to that had they not been displaced, this would indicate that these animals were using path integration or this vector-based navigation to find their way home. So here's a trial with an experiment that's just outlined for you in the cartoon. So you can see the animals leaving its burrow and will eventually locate the food. Okay, so the animals found the food, and now I'm displacing the animal along the track. And you can see from the homeward path of this animal that it's orienting towards the expected location of the burrow had the animal not been displaced, showing that it's using path integration to find its way home. So looking at the plot on the right, you can see the homeward paths when animals were displaced were oriented towards that expected location of the burrow rather than the actual burrow's location. And if I were to align all these starting points and also align the expected burrow's position, you can see that this like homeward path distribution doesn't look all that different from when animals were not displaced. So looking at these orientation plots, when animals were displaced, they were oriented towards the expected location of the burrow, the blue circle, rather than the burrow's actual location, represented by this red circle. So this showed they were using path integration to determine the direction of their home. But if you can recall from earlier, if an animal's path integrating, it also needs to know how far it should travel before reaching its goal. So in order to examine if mantis shrimp were able to do this, I compared the length of homeward paths before search behaviors were initiated. So those are the lengths of these red paths on the right to the beeline distance or the most direct path from the food location to the burrow. So looking at the box plot on the left, the y-axis is the percentage of the beeline distance traveled. So a value of 100% would indicate the animal went exactly that beeline distance before starting its search behavior. A value of 200% would indicate the animal went double that distance, 50 would be half. And we can see from both these groups that animals were able to go approximately that beeline distance from the food location to the burrow before starting their search. So this showed that these animals were using path integration to determine both the direction and the distances they should travel before reaching home. Okay, so now I've shown you that mantis shrimp are using path integration to locate their homes, and it's also likely that they're using the landmark when it's available. So in order to further verify that mantis shrimp are indeed using the landmark when it's there, and to see what would happen if when the landmark navigation and path integration systems were placed in conflict, I placed the track adjacent to the burrow and placed the landmark on the platform. So when the animal was away foraging from its burrow, I would displace this landmark to a new location in the arena. So here's an example of a trial in which I did this. Initially, the landmark was located here adjacent to the burrow, but while the animal was foraging, I displaced the landmark here. So you can see in this case, the animal oriented towards that displaced landmark and searched for its burrow along the side in relation to the landmark where it should be. However, this wasn't always the case. In this trial here, the animal completely ignored that displaced landmark and instead oriented towards its burrow's location. And when I did this with many animals, I saw behaviors all sorts in between. So many animals oriented towards that burrow. But what I often found was that animals would orient initially towards that displaced landmark, only to turn away from the landmark and orient towards the burrow instead. So that can be seen here. So this orientation plot is showing the initial home vector orientations, like I showed you before. So you can see some animals were oriented towards the landmark and others were oriented towards the burrow. Next, I measured the orientations of the search paths in relation to the burrow's location once they broke from that initial home vector trajectory. So that's represented by this row 2 here. So when I did that, I found that these initial search path orientations were oriented towards the burrow's location. So this is interesting for a few reasons. First, this indicates that the landmark navigation and path integration systems that mantis shrimp possess seem to be running in parallel. They're independent systems that are running in parallel. And second, it seems that the path integrator is continually running even during homeward travel. Okay. So I guess one thing that I've neglected to mention so far are the search behaviors that mantis shrimp exhibit once they run through their path integrator without finding their homes. So looking at this search pattern here, this is outlined in the path tracings here, and it's color-coded by time, where earlier parts in the search are in yellow, and the colors get cooler the longer into the search they get until they ultimately get to violet. So here's another search pattern displayed this way. And from this search, I think the strategy of mantis shrimp searches become a bit clearer. So during this trial, an animal carried its shell with its food in it along its home vector until it started its search behavior, at which point it dropped the shell. So it essentially left a marker for itself at that search initiation point. And here we can see that these searches appear to be comprised of these loops that start and end near the location of that search initiation point, and that these loops get larger over time. So here's the same search now color-coded per loop, and it becomes a bit clearer now. So you can see that the loops get larger and larger over time until eventually the animal finds its home. So I decided to plot the sizes of these searches over the course of time. And you can see each of these searches represented in different colors, all increase in size over the course of their search. However, they appear to be different sizes. So I wanted to explore how these mantis shrimp searches work, the rate of this mantis shrimp search expansion. So in order to do this, I normalized the initial sizes of these searches, and that can be seen here. So after normalizing the initial sizes of mantis shrimp searches, you can see that these searches seem to expand at a very similar rate. Now, optimal search theory predicts that searches such as these should expand proportionally to the square root of time. And that's due to error that is accumulated over the course of the search. So in order to explore how mantis shrimp searches expanded, I fit a power function to these data and found that mantis shrimp searches expanded in size proportionally to time to the 0.43. Now, this is actually quite similar to that predicted by optimal search theory, where it's expected that the sizes of searches should expand at time to the 0.5. So this indicates that mantis shrimp searches expand quite similarly to that predicted by optimal search theory. Okay. So, so far I've talked about the navigational strategies that mantis shrimp are using during navigation. Next, I want to talk about the sensory cues our mantis shrimp are using during path integration. Okay, so during path integration, an animal must monitor the directions it turns and the distances it travels. Focusing on the directions of turns, animals use all sorts of orientation cues. So a very prominent orientation cue that many animals use is the sun in the sky. So all sorts of animals will use this prominent cue for orientation. Another cue that many animals use is the celestial polarization patterns caused by the scattering of sunlight in the Earth's atmosphere. So this scattering creates a really predictable pattern that many animals that are sensitive to polarized light can use for orientation. Other animals will use this distinct panorama of their surroundings for orientation. Some animals are thought to use inertial cues for orientation, such as rotational accelerations estimated by the vestibular system of mammals. And other animals are even been shown to use geomagnetic fields for orientation. So mantis shrimp could use all sorts of potential cues for orientation. So in order to determine what kind of cues mantis shrimp were using, I decided to simplify things by trying to categorize if mantis shrimp were using allothetic or idiothetic cues for orientation. So an allothetic orientation cues are independent of self-motion, and they're often anchored to the world external to the animal's body. So example of allothetic orientation cues include celestial compasses like the sun in the sky, or the Earth's geomagnetic field for orientation. Now, the opposite of an allothetic orientation cue is an idiothetic one. Now, idiothetic orientation cues are manifested by self-motion, and they're often anchored to the animal's body. So a good example of an idiothetic orientation cue would be the proprioceptive compasses informed by the vestibular system in mammals. Okay, can everyone hear me? Okay, so in order to test the orientation cues that mantis shrimp are using, I built these circular orientation arenas. They're very similar to the navigation arenas that you saw before, but there are a few differences. First, there's a flat white base in these arenas, and in the center of the arenas, there was a platform that was rotatable. In the center of the platforms, I'd placed food. Like before, the burrow was set into the base of the arena, and this entire setup was placed outdoors under open skies in a wide open field away from any noticeable structures. So I would let the animals find the food in the center of the arena. Once the animals found the food, I would slowly rotate them 180 degrees. If animals were using allothetic cues for orientation, I predicted that they should orient back to the burrow despite their passive rotation, since those cues have not changed in relation to the burrow's location. However, if they're using an idiothetic cue for orientation, I predicted after a slow rotation, animals should orient in the opposite direction of their homes. I did this initially under two experimental conditions. The first, in which they were not manipulated. I let animals find the food and find their way home without manipulating them. Second, I slowly rotated the animals 180 degrees on that platform. I did this under 3 environmental conditions as well. The first under nice clear skies when the sun was clearly visible. So this is a full sky image taken with the fisheye lens camera. So you can see the sun's clear right here. Under the second environmental condition, I did this under partly cloudy skies when the sun was covered by clouds, but blue patches of sky were still visible. So you can see here the sun's covered, but there's all this blue sky available. These images on the right are polarization images calculated from these fisheye lens camera images I took. And you can see that redder areas or warmer areas in the images indicate places in the sky with a higher proportion of polarized light, while cooler areas indicate places in the sky with a lower percentage of polarized light. So you can see that there are these bands of polarized light that some animals can use for orientation, even when the sun is obscured. And the third environmental condition was under heavily overcast skies when the majority of celestial information was obscured. Okay, so here will be the results. So when animals were not manipulated, under clear skies, animals oriented towards their home. This is also true under partly cloudy skies when the sun was covered by clouds, as well as under heavily overcast skies. So under all experimental conditions, when animals were not manipulated, they oriented towards their burrows. Next, after rotating the animal 180 degrees, under clear skies, animals oriented towards their home despite their passive rotation. And this is also true under partly cloudy skies when the sun was covered by clouds. However, under heavily overcast skies, animals oriented in approximately the opposite direction of their homes after being rotated. So on all groups, animals are significantly oriented towards their burrow, except for when animals were rotated under partly cloudy skies. Those animals were oriented away from their homes. And from these results, we can learn a few things. First, that mantis shrimp appear to possess allothetic compass cues, and these cues are celestial in nature. So they may be using the sun for orientation, but as can be seen from this second condition, when the sun was covered by clouds, as long as patches of blue sky were visible, animals were still able to orient correctly despite their rotation. So under this condition, they're likely using other celestial cues than the sun for orientation. It also appears that mantis shrimp possess or monitor idiothetic information for orientation when all celestial information is obscured. Since under the condition when they're rotated under heavily overcast skies, they're oriented in approximately the opposite direction of their homes. If they were unable to do this, you would expect random orientation in this condition. Okay, so since I've shown that they're using celestial cues for orientation, I next wanted to understand which specific celestial cues they were using. Since the sun's the most prominent cue in the sky when it's available, I wanted to determine if mantis shrimp were orienting using the sun. So mantis shrimp are most active during dawn and dusk. My species are almost exclusively active at those times. So during those times, the sun's at a fairly low or fairly close to the horizon. So in order to determine if mantis shrimp are orienting using the sun, I used a very classic sun displacement experiment in which I shielded the arena from the sun with a board, and I used a mirror to reflect the sun to the opposite side of the arena from where the sun actually is. So for an animal in the arena, it would appear that the sun is on the opposite side of the sky from where it actually should be. So again, I waited for the animal to find the food. Once it found the food, I would mirror the sun. If they were using the sun for orientation, they should orient in the opposite direction of their burrows. So here's an example of that experiment. All right, so once the animal finds the food in the center, the sun is shielded and mirrored. And you see in this condition, the animal orients in the opposite direction of its home. So looking at the data as a whole, when the sun was blocked by the board in isolation, mantis shrimp were still able to orient towards their burrow. However, when the sun was also mirrored in addition to being blocked by the sun, the majority of animals oriented in the opposite direction of their home. So this showed that mantis shrimp were orienting using the sun. Okay, but as I told you before, it was clear that mantis shrimp weren't exclusively using the sun. They're also relying on other celestial information for orientation. So I wanted to understand if mantis shrimp were orienting using celestial polarization patterns. In order to do this, I brought those same arenas I used outdoors into a dark room, and I surrounded these these arenas with a curtain to create a homogeneous environment. I placed a light source directly over the arena and placed a polarizing filter between the light source and the arena. So this created a polarized target for an animal directly overhead it. I would let the animal find the food. Once the animal found the food, I would rotate that polarizer 90 degrees. Since polarization information in isolation is bidirectionally ambiguous, I predicted that animals should orient perpendicular to the direction of the burrow. So here's an example of that experiment. Okay, so the animal found the food and I've rotated the polarizer 90 degrees. And you can see the animal orients perpendicular to the direction of their home. So looking at the data as a whole, when the filter remained fixed, mantis shrimp either oriented towards or away from their burrow. However, when I rotated the polarizer 90 degrees, mantis shrimp oriented perpendicular to the direction of their burrow, showing they were using that overhead polarization stimulus for orientation. So with all these orientation experiments, I could propose a hierarchy of compass cues these animals are using for orientation. So when mantis shrimp are most active, the majority of animals appear to orient using the solar azimuth for orientation. However, when it's obscured, as long as patches of blue sky are visible, mantis shrimp appear to orient using celestial polarization patterns and potentially other celestial cues I did not test. And finally, at least in my arenas, when all celestial information is obscured, mantis shrimp appear to orient relying on idiothetic cues for orientation. Okay, so to summarize what I've told you so far, mantis shrimp navigate home using a navigational strategy called path integration, and they do so by orienting using the sun, overhead polarization patterns, and idiothetic cues. However, path integration is not perfect, and it often leads them not directly to their burrows. So when this is the case, mantis shrimp enact these stereotype search patterns that are composed of loops that expand in size, similar to that predicted by optimal search theory. Finally, if landmarks are available, they will use them in parallel with their path integration system to accurately pinpoint the location of their homes. Okay, so that's all I have for you today. So I'd just like to thank a few people. First, I'd like to thank the Cronin Lab, especially my graduate advisor, Tom Cronin, for a really wonderful graduate experience. I'd also like to thank Natalie Roberts, John Park, and Sam Hulse for research help, as well as Keith Berkman for showing me where mantis shrimp can be found in the field. I'd also like to thank John Cohen and his students at the University of Delaware's College of Earth, Ocean, and Environment. He hosted me while I did all that orientation experiment. And I'd like to also thank my graduate committee and my funding sources. All right, thanks, and I can take any questions.
Thank you so much, Ricky. That was really beautiful, beautiful work. I will hand it over to Sarah. We have about 3 minutes for questions.
Great, thank you, Ricky. And our first question is from Barbara Webb.
Hi, Nick. Hi, Ricky. Can you hear me?
I can hear you, yes.
Great. Okay. I actually have several questions, but I'll go with my first one. When you did the Q conflict experiment where you'd move the landmark, you described them as going towards one or towards the other, but it seemed like, at least in the initial direction, so at least some that were going in between. Did you not think that happened, or is that just due to just averaging two directions they were going?
So it's kind of hard to say because if you're looking at the orientation plots, I can probably go back to them. The amount of error that they generally have during their homeward orientation kind of encompassed the directions of the landmarks. That's kind of far back, sorry. Let's see. Oh, here we go. So it appeared that there were some that were halfway in between the direction of the landmark and the burrow, but it's really difficult for me to understand if that was like a kind of like averaging of the two expected directions, or if it was one or the other.
All right. Thanks. I'll let some other people ask, and if I get a chance, I'll ask my other question.
Okay.
All right, next is Malcolm.
Yeah, thanks. That was great. And I actually had a similar question to Barb's. And I guess it looks to me like these animals are pretty good Bayesians. They are estimating the uncertainty of the cues that they have available to them. And I'm curious, that's pretty apparent in the celestial navigation experiments that you did that were so elegant. It's less clear to me what cues they're using with respect to landmark displacement to sort of say, okay, that could be like a piece of weeds or something that just got tumbled by the current. So I'm not going to really count on that. I'm going to use my path integrator instead. So do you have any sense as to what the animal is using to discount either either path integration or landmark cue?
Yeah, that's a good question. So during the landmark displacement experiments, I tried to determine what they might be using to either trust the landmark or the burrow. I found that nothing really was clear to me. So there was a few ideas I had. One, that perhaps there was a bit of a range at which I was able to displace the landmark. And I thought maybe if the discrepancy between the burrow's location and the landmark's location was large enough, they would ignore the landmark. But that didn't appear to be the case. There wasn't very strong evidence for that at all. Another idea I had was that since during path integration, these animals are monitoring the directions they travel and the distances they travel as well. And those measurements have a bit of error associated with them. So over the course of time, these errors accumulate. So therefore, with longer outward paths, mantis shrimp actually exhibit a larger homeward error. So I didn't have time to talk about that, but I found that to be the case. So I thought that perhaps animals with longer outward paths would tend to trust the landmark since their path integrator was a little less accurate. But when I did that analysis, it wasn't significant, but I think the sample size was a little too low to completely discount that hypothesis either. So I'm not really sure how they do that. And then as far as your question about the actual constancy of the landmark itself, perhaps they have some sort of way of evaluating their surroundings to know what is a reliable landmark and what isn't. I'm not exactly sure.
Okay, great, thanks.
I'm going to break in here. That's our time. I know there's a lot of other questions, and so you can message Ricky and you can chat about those directly. Thank you very much for a wonderful talk, a second wonderful talk. Really appreciate it. Okay, great.